பயாலஜி இன் தமிழ் கோர்வை சேனல் சார்பாக அனைவருக்கும் வணக்கம். என் பெயர் கண்ணதாசன். In this video I am going to teach you the lesson Taxonomy and Systematic Botany part 4. So video 1, video 2, video 3 already published, and the link for those video given in the description. You can make utilize of it. This lesson is the lesson 5, volume 1, 11th standard Botany State Board book, chapter 1, 11th standard CBSE book. The units that we are going to learn are 5.7 Herbarium, preparation and uses, 5.8 classification of plants, 5.9 need for classification, and Botanical Survey of India. This is one of the box information. Now I request all the students to take the textbook, go to the particular page, and hints taking notebook along with pen. These are the very essential materials in order to attend the video. When you are taking the notes, it will be very useful during your revision time towards your examination. You need not go through the entire book. Just go through the hints will help you to speed up your learning process at the time of revision. This video also going to be accompanied by one Google Doc form question paper which carries 20 MCQ. It is worth of attending the 20 MCQ. You will be asked to give your email ID as well as WhatsApp number so that you will be allowed to attend the MCQ, and exclusively preparing for the board student, not preparing for NEET sir. Even if you can attend the MCQ as well as 2 mark will be there, 2 3 mark and 1 5 mark. You can write it in the separate notebook and do self correction. I hope this video will be very useful for you. If you like this video, please press the like button, share this video to all your friends, comment on it, subscribe our Biology in Tamil Korvai channel, and also press the bell icon to receive the notification. I wish all the students to get full mark in the Google Doc form question paper. In order to get full mark, you please stay with the video till the end to grasp all the concepts. Now let us enter into this video, students. Herbarium first. Herbarium, it can be asked, write note on herbarium, can be asked in 2 mark question. Herbaria are storehouses of preserved plant collection. So this is one of the collected plant, pressed, dried and mounted on herbarium sheet along with one label. So the entire herbarium process is almost over. So here you see plants are preserved in the form of pressed and dried specimen mounted on a sheet of paper. Herbaria act as a center for research. Center for research. Here, how this herbarium sheet can act as a center for research, sir? You may ask one question. Herbarium, not only referring herbarium singular, herbarium plural. Herbarium not only referring the herbarium sheet alone, individual plant, but it also refers the institute where all these herbarium sheets are properly maintained for a very long period of time, like Royal Botanical Garden. That is also called as herbarium. So herbarium acts as a center for research. Definitely all the taxonomists, scientists, industrialists, those who want to do or produce certain substances, very important substances, nutrients from the plants, they want to identify the plant. At that time they are in the need of the herbarium to identify the correct plant. Even a small mistake in the identification of the plant may lead to the major failure in their task. So this is the importance of herbarium. So it is a source of material for various systematic work. It means the classification work. Now let us go learn about herbarium, preparation and uses. Herbarium definition. So this is the herbarium where you can see the racks where the herbarium sheets are maintained. Herbarium specimen is defined as a pressed and dried plant sample that is permanently glued by using the adhesives, glued or strapped to a sheet of paper along with a documentation label. So this is the definition for herbarium. This can be asked in 2 mark question. Preparation of herbarium specimen includes following steps. So this can be asked in 3 mark question because it consists of 6 steps. By writing each step you can get 1.5 mark. First one is plant collection. Under plant collection there are 4 types given. One is field collection, second one is liquid preserved collection, third one is living collection, and the last one collection for molecular studies. So this is one step, it is plant collection. Second step, documentation of field site data. Third step, preparation of plant specimen. Fourth, mounting herbarium specimen. Fifth one, herbarium labels. Sixth one, protection of herbarium sheets against mold and insects. So these are the 6 steps, can be asked in 3 mark question. Please take note of it in your notebook. Plant collection, in which the first one is field collection. We are going to the actual field. The field here, it is not only referring the cropland, but also referring the even the forest, grassland, where we are going to collect the plants, that is from its actual native place, that is called as field collection, right? Suppose we are going to Amazon forest collecting the plant, that is called as field collection. Collection for molecular studies, second one. There are so many molecular studies going on with respect to the plants because they consist of essential molecules which are very useful to the human life. So, in order to separate the molecules from the plants, they are collected. And also make it very clear, the DNA, RNA, proteins, these molecules are very much important for classification. Classification, according to the phylogenetic principle, phylogenetic relationship can be established with the help of molecules. So the molecular separation should be done so that the collection should be made. That is called as collection for molecular studies. Third one is liquid preserved collection. This is actually the information collected from the reference. The plants are preserved in the liquid, certain solution. In one of the reference solution, it is given Copenhagen solution. What is Copenhagen? If you ask that question, it is actually referring capital of Denmark. In that place, there is one laboratory. From there, it is produced first. So it is called as Copenhagen solution. The composition of the Copenhagen solution: 70% of industrial methylated spirit. The spirit is used as a preservative. 28% distilled water and 2% glycerol. The glycerol prevent the specimen from becoming too brittle because of the preservative. So the flexibility should be there for the specimen, that is provided by the glycerol. But nowadays the formaldehyde is not used. Suppose you are going to the laboratory and asking your teacher what is the solution used as a preservative. Usually we say formaldehyde, because all those specimens are preserved about 20 years back, 25 years back, still it is there. Very good collection. Okay, so in those days we were using formaldehyde, but nowadays due to the formaldehyde causing lot of health risk, so we are not using formaldehyde, right? In Royal Botanical Garden, they are straight away using 70% ethyl alcohol or 70% methylated spirit or denatured alcohol with 30% water. That's all. From my reference, I am able to get this from their website. Rubber seals in order to cap the bottle, and here you can see the glass lid is used to seal this jar, so that the operation of this preservative can be prevented. Very clear. And also periodically we have to check the fluid level so that the plant part are not exposed outside the solution level. Right. Now plant collection, the last one is living collection. Example here I am giving, this is the Royal Botanical Garden, but I am giving Australian National Botanical Garden. So Botanical Garden, you know very well, carries lot of plants, live plants. So that is the reason why it is called as living collection. Maintains a living collection of around 74,000 individual plants arranged in 40 hectares of themed gardens. Okay, so this is called as living collection. So the first one, step 1, plant collection is over. Plant specimen with a flower or fruit is collected like this. Step 2, documentation of field site data. Documentation of field site data. Certain data ought to be recorded at the time of plant collection. That is very important. At the time of plant collection, whatever the data we collect, that is extremely important till the end of the herbarium sheet life. The label is maintained. That information actually from this data. You can see the people who are collecting the plants having their notebook. That is the reason why you are asked to take the notebook and take note of it. Whenever you see a video or listen to your teacher's class, if you take the notes, it will be very important documentation, important record, which you cannot forget at all. So here also they are collecting the all information. What are the information they are collecting? Date, time, country, state, city, specific locality information from where the plants are collected. Latitude, the latitude on the imaginary line on earth, that is called as latitude, which is parallel to the equator. Latitude. Longitude, right? Longitude, one which connects both the poles, north pole and south pole. Longitude. Elevation, altitude, from mean sea level, what is its height, from where it is collected. Elevation. Landmark information, all these data are required. Suppose if we want to revisit the plant, all these informations are required. These data will be typed onto a herbarium label. Step 3, plant specimen collected from field is pressed immediately. Immediately it is pressed before it is undergoing evaporation, right? Desiccation. It is immediately pressed with the help of portable where the plant press, the wooden plant press which we are able to carry along with us for field collection is called as portable field plant press. Then it will be transferred to the standard plant press. Plant specimen is transferred to a standard plant press. You see here, the size of the plant press is 12 inches into 18 inches. 12 inches into 18 inches, which between two outer 12 inches and 18 inches frame and secured by two straps. Very clear? So this is the way in which it is manufactured. The standard size of herbarium sheet. Now we are going to learn about step 4, mounting herbarium specimen. Once the plants are completely dried with the help of the plant press, but every day we have to keep the plant in that is sunlight and change the paper. Then every day evening we have to tie it up with the help of plant press. Not once it is that is tied, and finally after a month if you are taking it, no plant will be there, right? So the after it is completely dried, it is going to the step 4. It is called as mounting herbarium specimen. It needs herbarium sheet. The sheet size is also very standard, internationally accepted size. It is 29 centimeter into 41 centimeter. 29 centimeter into 41 centimeter. Specimens are affixed to herbarium sheet with standard white glue or solution of methyl cellulose. So this can be used as adhesives. Step 5, herbarium label. Herbarium label is very, very important because it furnishes all the details about the plant collection. Even the person who collected the plant, its size also mentioned. So plant press size is mentioned, herbarium sheet size is mentioned, and here the herbarium label size is mentioned. It is 4 to 5 inches wide and 2 to 3 inches tall. 4 to 5 inches wide and 2 to 3 inches tall. Here the tall is less, width is more. So you must be very careful about this information. Whenever it is asked in the question tall, 4 into 5, 4 to 5 we will take. So please take note of it. A typical label contains all information like, so what are the information should be furnished in the herbarium label? 3 more question. Habit of the plant, habitat, from where it is collected, vegetation type, whether it is a tree or grassland, etc. Landmark information, already we'll discussed latitude, longitude, altitude, etc. Image document, collection number. As we are collecting many plants in a day, we have to number it along with the date. Date of collection, you see, along with the date and name of the collector. Step 6, protection. We have to protect the herbarium sheet for a very longer period of time. For more than 100 years in Royal Botanical Garden, the herbarium sheets are well preserved. How? By using various preservatives like cation of 2% mercury chloride, DDT, naphthalene, carbon disulfide, and also fumigation using formaldehyde. Fumigating the entire laboratory. Okay, where the herbarium sheets are maintained by using formaldehyde. Presently, deep freezing minus 20 degrees Celsius. Deep freezing the plant materials at minus 20 degrees Celsius. This method is followed throughout the world nowadays. And this is one of the small box information about world's smallest water lily. Smallest water lily. Its scientific name is Nymphaea thermarum. Nymphaea thermarum was saved from extinction. So once it was endangered species, and it is saved from extinction when it was grown from seed at Kew, England, London, Royal Botanical Garden. Please recollect the information. In the year 2009. Now we are going to learn about uses of herbarium. This is one of the 5-mark question. First one is herbarium provides resource material for systematic research and studies. Resource material. Whomever want to do some studies in relation to a plant, immediately they have to go and identify whether they have selected a correct or identified a correct species or not. Then only they can be successful. Second one, it is a place for orderly arrangement of voucher specimens. Here they are using one new word called as voucher specimen. That alone can be asked. Write note on voucher specimen in 3 more question. Point 2, 3, 4 are about voucher specimens. Voucher specimens serve as a reference for comparing doubtful, newly collected, fresh specimens. We are going to a wild environment, assume, where we are identifying one new plant. According to us, it seems to be a very new plant, but it look like it's another plant. So doubt arise. Understood? So we are going to the herbarium and we are asking for the voucher specimen. We are comparing it with the voucher specimen along with our collected specimen. Then we are coming to the conclusion whether it is a new plant or it is a already discovered voucher specimen. Okay, already discovered plant. Voucher specimen play a role in studies like floristic diversity. Studies like floristic diversity. Diversity we want to study in a particular country. Environmental assessment, ecological mechanism and survey of unexplored areas. Survey of unexplored areas. So far that area is not explored by human being. we are able to go to that particular area and collecting new plants and comparing, as I already told you, with the voucher specimen, so that we can say, yes, I discovered this plant. Are you clear? So this is the use of voucher plants. Okay, herbarium provides opportunity for provides opportunity for documenting biodiversity. It means what are the plants available in Tamil Nadu, what are the trees, what are the shrubs, how many of them are commercially important. All this called as documentation. And studies related to the field ecology and conservation biology. Suppose we are identifying a plant, it is very rare. So naturally we will label it as endangered species and we will go for conservation. What is a voucher and why is it important in research? This is the reference information from my side. Okay, so as a preserved specimen of an identified taxon deposited in a permanent and accessible storage facility. So that is a voucher specimen. It serves as a supporting material for published studies of the taxon. About that particular voucher specimen, there will be definitely published information, published books will be there. So it supports that published information. They are very crucial in authenticating the taxonomy of an organism. Authenticating, authenticity, authoritatively it says, yes, this is the right plant, this is the particular species. So as a tool for identifying localities of the taxon and for additional taxonomic, genetic, ecological and environmental research. So it allows all this research work to be carried out with respect to a particular species. Are you clear? Now we are going to learn exclusively about Kew Herbarium. 5 more question. Kew Garden is situated in southwest London that houses the largest and most diverse botanical and mycological fungal collections in the world. Founded in the year 1840, living plant includes more than 30,000 different kinds of plants. Unimaginable. 30,000 different kinds of plants, living plants. While herbarium, which is one of the largest in the world, the Royal Botanical Garden is the largest in the world, has over 7 million preserved plant specimens. One of the largest, please take note of it. The library contains more than 750,000 volumes and the illustrations. It means illustrations of plants, picture, and also a collection of more than 1,75,000 prints, books, photographs, letters, manuscripts, periodicals, maps, and botanical illustrations. So all these are available where? In Royal Botanical Garden, Kew, England. International Herbarium. Here there are 4 herbarium given, so it can be asked in 5 more question. There are more number of numerical data also. The first one is Museum National d'Histoire Naturelle, Paris, France. Year established 1635. Acronym P or PC. Number of specimen. You see here, I think it is 1 crore, 100 lakhs. Yes, 1 crore. New York Botanical Garden, second one. Bronx, New York, USA. Established in the year 1891. Acronym capital N, capital Y. Here 72 lakh specimens. Third one is Komarov Botanical Institute, St. Petersburg, Leningrad, Russia. 1823, that is the year of establishment. And its acronym is capital L, capital E. 71 lakh 60,000 species. Fourth one is Royal Botanical Garden, Kew, England, UK, 1841, the year of establishment. Acronym K, K stands for Q. Number of specimen, 70 lakhs. So the Kew, England, okay, this is one of the largest herbarium in the world. National Herbarium. What about our country? You may ask. Madras Herbarium, BSI, Botanical Survey of India, Campus, Coimbatore, 1955. Acronym MH. Number of specimen 4,08,776 plants. Central National Herbarium, West Bengal, 1795. CAL is its acronym. 2 lakh specimens. Jawaharlal Nehru Tropical Botanical Garden and Research Institute, Trivandrum, Kerala, 1979. Its acronym is TBGT, 30,500 species. Then fourth one is in Chennai, Presidency College Herbarium, Chennai, 1844. 1844. The acronym is PCM and number of specimens 15,000 specimens. So this can also be asked in 1 5-mark question, students. 5.8 Classification of plants. Imagine walking into a library and they are here giving some imagination that is narration of a story. Okay, they are telling it. Imagine walking into a library and looking for a Harry Potter storybook. You are looking for a Harry Potter storybook. Okay, as you walk into the library, you notice that it is under renovation. Now all books are scattered. So they are creating an imagination that it is very difficult for you to identify Harry Potter storybook. Very clear, because all the books are scattered. Will it not be hard to find the exact book you are looking for? It might take hours together. So you decide to come the next day when all the books are arranged according to the genres. Genres, it means according to the categories or subject. They are now arranged perfectly. One rack for adventure, another for detective, fantasy, horror, encyclopedia, and so on. It is arranged. Now, what is your condition? You automatically know Harry Potter is in the fantasy section, and it takes less than 10 minutes for you to identify the particular book. That is because the books have been classified. So now you have to imagine the books along with the plant specimen. The plant specimens are scattered in the entire world. Very clear. So by classification, we are bringing it in a proper order, as if the books are arranged in the library. That is the way in which we have to compare these two situations. Similarly, there is a vast assemblage of group of plants in the world. They are scattered in a different place. We are identifying it, describing it, naming it, then classifying it, so that we are able to easily access them for various purposes. It is possible to study and understand all of this? No, it's not possible. Since it is difficult to study all these plants together, it is necessary to devise some means to make this possible. So that is the reason why classification in a particular family, one important plant, when we are studying as a typical plant, that is most of the character applicable to the all the plants of the particular family. That is the way in which we are able to access and study the entire plant kingdom. Okay, next. Classification is essential to biology because there is a vast diversity of organisms to sort out and compare. Unless they are organized into manageable categories, it will be difficult for us for identification. Biological classification are the inventions of biologists. These are inventions of the biologists based upon the best evidence available. So definition for classification. The scientific basis for cataloging and retrieving information. Cataloging and retrieving information. We are cataloging it, retrieving it. Whenever we want, we can go and get it back. That is called as retrieving. Information about the tremendous diversity of flora is known as classification. So this can be asked in 1 2 more questions, students. Classification paves way for the arrangement of organisms into groups on the basis of their similarities, dissimilarities, and relationship. Now you take angiosperm, dicotyledons and monocotyledons. Dicotyledon consists of 2 cotyledon. So that is similarity. Monocotyledon consists of 1 cotyledon. So a group of plants are going into this category. The purpose of classification is to provide a systematic arrangement expressing the relationship. What is the relationship between the two plants? Now, through the molecular studies, they are able to establish their phylogenetic relationship. Even the fossils are not required. To that extent, the molecular studies and molecular classification becoming one of the very famous development in the field of taxonomy. Taxonomical ranks. Taxonomists have assigned a method of classifying organisms which are called as ranks. These taxonomical ranks are hierarchical, like uh, starting from species, genus, family, order, class. Likewise, it goes, that is called as hierarchical. The scheme of classification has to be flexible, allowing so that it allows the newly discovered living organism to be added. Okay, where they fit best, fit best. It means the suitable characters they are having with the other groups. Need for classification. This is one of the 3 mark question because it consists of 3 to 4 points. 1. Understanding the classification of organism can give an insight into other field of science, and a significant practical value. Classification helps us to know about different taxa, their phylogenetic relationship. That is very, very important. Monocotyledon evolved first or dicotyledon evolved first? We are able to answer. That is called as phylogenetic relationship. And the exact position. It helps to train the student of plant sciences with regard to the diversity of organism, plant diversity, so that we are able to understand the plants properly and their relationship with other biological branches. So these are the need for classification. Here we are going to learn about Botanical Survey of India. This can be asked in one 5 mark question. On 13th February 1819, a survey was formally constituted and designated as the Botanical Survey of India in order to document all the plants in India. After independence, the need was felt for a more comprehensive documentation of the country's plant resources to boost the economy. The economy of the country is closely related to biology students. Remember that. Padma Dr. E.K. Janaki Ammal was appointed as officer on special duty on 14th October 1952. Then reorganization plan was finally approved by the government of India on 29th March 1954 with Calcutta as the headquarters of Botanical Survey of India. Jammu Tawi Botanical Garden has been named after Dr. E.K. Janaki Ammal. Okay, so these are the information about Botanical Survey of India. So this is the end of this video, student. So far we learned 2 point. Thank you very much.